Reproduction in OEDOGONIUM. In OEDOGONIUM, sexual reproduction is of advanced rugamous type. Sex organs are well developed. Male sex organ is known as antheridium and female sex organ is known as oogonium. Antheridium produces male gamete known as antherozoids and oogonium produces female gamete that is known as egg. So now let us discuss about the process of sexual reproduction in OEDOGONIUM. All the species of OEDOGONIUM can be divided into two different groups on the basis of distribution of sex organ. One is macrandrous species, other is nonandrous species. In macrandrous species, the antheridia are born on normal size filament, whereas in nonandrous species, antheridia are born on short filament known as dwarf male or nonandrium. Macrandrous species can be monoecious or dioecious. In monoecious species, the antheridia and oogonia are born on the same filament, whereas in dioecious species, antheridia and oogonia are born on different filament. Second is nonandrous species. Nonandrous species is divided into two different groups. One is gynandrosporous species, other is ideoandrosporous species. In gynandrosporous species, the androsporangia and oogonia are born on the same filament, and in ideoandrosporous species, androsporangia and oogonia are born on different filament. So now we will discuss about the development of antheridium, oogonium and also about androsporangia. So first is the development of antheridium. Any vegetative cell having apical cap can behave as antheridial mother cell. This is the antheridial mother cell. This cell divides transversely to form a small cell, that is the young antheridium, and a larger cell that is known as sister cell. The sister cell divides repeatedly to form a number of antheridia, and the nucleus inside each antheridia divide mitotically to form two daughter nuclei, and each of these daughter nuclei undergo metamorphosis to form a male gamete known as antherozoids. The antherozoids are uninucleate and multiflagellate structure. They are released in a vesicle, and the vesicle later on dissolve, thereby releasing the antherozoids so that they can participate in the process of fertilization. So inside each antheridia, two antherozoids are formed, and the antherozoids are uninucleate and multiflagellate structure. This is about the development of antheridium. The process of antheridium is very simple in macrandrous species. In macrandrous species, antheridia are born on normal size filament, whereas in nonandrous species, the development of antheridium occur on short filament that is known as dwarf male or nonandrium. So in nonandrous species, specific structures are formed known as androsporangia. The process of development of androsporangia is similar to the process of development of antheridia. The only difference is that inside each androsporangium, a single androspore is formed, whereas in each antheridia, two male gametes are formed. So, any vegetative cap cell can behave as androsporangial mother cell, which can divide to form a number of androsporangia. And inside each androsporangium, a single androspore is formed. The androspore is a uninucleate and multiflagellate structure, and this androspore is released in a vesicle. Later on, the vesicle wall dissolves so that the androspore is released to the outside. So this androspore move to the nearby oogonium. This is the oogonium and this is the supporting cell. This androspore move and attach with the supporting cell. There the androspore undergoes germination to form a short filament that is known as dwarf male. So this dwarf male is parasitic on this filament. This dwarf male is also known as nanandrium. In nanandrium, transverse division occur several times. So many antheridia are formed. These are antheridia. Inside each antheridium, two male gametes or antherozoids are formed. Later on, the antherozoids are released to the outside so that they can participate in the process of fertilization. This is about the development of antheridium in nanandrous species. Next is oogonium. Oogonium is the female sex organ. Any vegetative cell that has apical cap can behave as oogonial mother cell. This is the oogonial mother cell. The oogonial mother cell divide transversely to form a oogonium and a lower supporting cell. The lower supporting cell can also behave as oogonial mother cell and divide again so that few oogonia can be seen as present in a row. This oogonium is a globose structure that has a transverse slit or pore. Nearer to the slit there is a colorless area that is known as receptive spot. And the oogonium contains a single nucleus. And this is the female gamete. This is about development of antheridium and oogonium, and also about the development of androsporangia, androspore and antherozoids. Next is fertilization. The process of fertilization occurs between male gamete and female gamete. Here you see the male gametes are released to the outside. They move closer to the oogonium and the antherozoid enter into the oogonium through the slit or pore which may be present. So many antherozoids may enter into the oogonium, but only one antherozoid will fuse with the female nuclei that is present inside the oogonium. So fusion occurs between female nucleus and male nucleus inside the oogonium. Fertilization occurs inside the oogonium. Here the female nuclei and male nuclei fuse together to form a diploid structure known as zygote. And this is the wall of the oogonium. Later on, the oogonial wall rupture and the zygote is released to the outside. This zygote secretes a three-layered wall around it. After this zygote undergoes a period of rest and settle on a suitable substratum. Next, when the condition becomes favorable, this zygote germinates. During germination of zygote, the diploid nucleus divides meiotically to form 4 haploid nuclei. Each of these 4 haploid nuclei undergo metamorphosis to form a uninucleate multiflagellate zoomeospore. So inside each zygote, 4 zoomeospores are formed, and this zoomeospore undergo germination when the condition is favorable. So this zoomeospore settle on a suitable substratum in which the interior end is pointing downwards. The flagella are withdrawn and the zoomeospore elongate considerably. Following this, it divides transversely to form a apical cell and a basal cell. This upper cell, which is green in color, divide again and again to form a young filament, and the basal cell give rise to the holdfast. This is about the process of sexual reproduction in Odogonium.